Okay, hello everybody. My name is Brody Duncan. I'm a medical microbiologist here in Hamilton and an assistant professor in the Department of Pathology. So I was asked to do this lecture for medical microbiology for medical students. The topic of today's lecture is gram-positive cocci, but it is just going to be one in a series of lectures where we talk about different organisms and aspects of medical microbiology. The focus of this lecture is going to be for medical students that are mainly in pre-clerkship, but this is a topic where I found from my own training that seeing it again and again in different settings, for example, when you're first starting clerkship, when you're at the end of clerkship and doing some studying, even when you're a resident starting PGY1, as you're able to connect some of our teaching up with actual patients that you've seen and infections that you've diagnosed and treated, I think you'll be able to get more out of this. But hopefully this works as a good introduction to gram-positive cocci. In terms of our goals for today, so first we're going to learn the names of medically important gram-positive cocci. I find that sometimes with these lectures, people really teach you every single thing about every single organism, and then you get overwhelmed and it's hard to put it into context. So our main goal for today really is just to learn the names of these organisms. Once you know the names, then you can actually start to put them in some sort of like coherent context for when you're going to eventually be seeing and treating patients. So our next most important goal is to understand the relative virulence of each species. And then finally, we're going to talk about some very basic phenotypic tests. I think that, there's a lot of textbooks and videos talking about medical microbiology where they go into the details of these phenotypic tests. But if you're going to be a clinician where you're just on the wards and not actually setting foot in the lab, I think that's a waste of your time. So we'll just try and focus on the basics that you need to know at this stage of your training in medical school. So this is a slide that we had from our very first introductory lecture. So today we'll be focusing here on the left with our gram-positive cocci. You can see we have three main genera, Staph, Strep, and Enterococcus, and that's what we're going to be focusing on today. So gram-positive cocci are gram-positive, which as we've discussed means they're purple, and they're cocci, which means they're circles. For GPCs, they are a bit unique from other bacteria in that the arrangement of how the cells are on the slide is actually pretty useful and important information as well. So we often describe gram-positive cocci as being either in groups or clusters. So that's our picture on the left where you can see these little purple circles are just kind of scattered around and often in little bunches. Or on the right, we have gram-positive cocci in pairs and chains, where instead of being clustered together, you can see that as they divide, they actually form these chains or strips that have a bit of directionality to them. The reason this is important is because the most medically important gram-positive cocci actually are easily separated by these gram stain characteristics. So on the left, Staphylococcus are species that arrange in gram-positive cocci in groups. And on the right, you can see that Streptococcus and Enterococcus are gram-positive cocci that arrange themselves in pairs or chains. And they very reliably do this. This is the sort of thing where the technologist calling you in the middle of the night because Mrs. Smith has a positive blood culture can tell you right on the phone, oh, Mrs. Smith has gram-positive cocci in groups in her blood culture. And then you can hang in that phone knowing right away that it's probably some sort of Staphylococcus species. I will say there's a few other very uncommon bacteria that could be gram-positive cocci as well, but these are really the three genera that you guys need to know. In terms of a little mnemonic to try and help you remember, because I always struggled with this, Staphylococcus, from Greek or whatever it is it's derived from, means grapes. So you can think of a bunch of grapes. Sometimes when I was a medical student, also would think of just like stuff, like staph is a bunch of stuff like in a group or a cluster. Contrast this with Streptococcus, where the original name is from a chain, but you can think of it like a strip. So like a strip of cocci in a row is how you remember Streptococcus. Enterococcus, I have not come up with any sort of mnemonic for. They used to be a type of Streptococcus and then they got split apart. So you just kind of have to really jam pack that into your brain. Staphylococcus is in groups and Streptococcus and Enterococcus are in pairs and chains. So first we'll talk about Staphylococcus as a genus. So again, this is GPCs in groups. Staph aureus is the #1 organism we need to think about when we're thinking about Staphylococcus. And the reason for that is it is a nasty guy. Staph aureus is not your friend. It unfortunately is often part of our normal flora. The most common place it lives is in the nose, but it can also be on your skin, in your mouth, in your gut. But the thing about Staph aureus is that even though it can be part of the normal flora, it is a pretty virulent organism. It is aggressive. It causes cellulitis, where it doesn't just sit on top of the skin, but buries under and causes infections. It can also cause other infections like endocarditis, where it gets in your blood and then it sticks onto your heart valve and causes a really nasty and life-threatening infection. I don't have time today to talk about all the different types of infections that Staph aureus or any of these bacteria cause, but you need to understand that this is a virulent organism. When you see Staph aureus in a culture for your patient and it makes sense with the clinical syndrome, you should probably assume that it is a real pathogen that needs to be treated. I'm going to contrast that with coagulase negative staph. I'll talk about what coagulase means in a minute, but this is essentially a group of staph that are much less virulent, much less aggressive than Staph aureus. So they're part of the normal flora. It's totally fine and normal to have them on your skin, in your mouth. It's just when they get where they're not supposed to that you run into trouble. There's a huge long list of these coagulase negative staph, which is why they're kind of grouped together under this heading of coagulase negative staph. You don't really have to know the names of them, but in general, their names match up with things that don't sound super aggressive. Staph epidermidis on your skin, Staph hominis, very common in humans, Staph capitis, on your head. There's so many of these, you really don't have to memorize them. The one thing I will say is there's this Staph saprophyticus that for whatever reason is able to cause urinary tract infections, especially in young, healthy women. But on the whole, if you're seeing any of these organisms in cultures, they're usually not very aggressive. The most common place you'll see it is actually as a contaminant in a blood culture. As I said, because they live on our skin, if you have a nurse or a phlebotomist that's taking a blood specimen, you can imagine that if a little bit of the needle or the blood touches the outside of the skin before it goes in the tube, you could get a little bit of these coagulase negative staph on that blood in the tube, even if it's not actually in the patient. So that is probably the most common situation you see coagulase negative staph is as a contaminant, where it's just because the technique wasn't 100% there and a little bit of the skin bacteria got in the tube on the way to the laboratory. However, they actually can cause true infections as well. For example, if you have a PICC line or another sort of IV line that is a permanent connection between the outside skin and the blood inside, you can imagine that it's not that difficult for Staph epidermidis, for example, to slip in the top of the catheter and then worm its way down until it's exposed to the blood. And then once it's in the blood, it can actually cause a true infection. So again, just to emphasize, coagulase negative staph are not terribly nasty, but they can cause true infections, mainly when they go where they're not supposed to, which is the sterile sites of the body. I'll talk very quickly about what is coagulase if we're using it, as a descriptor for all these different types of Staphylococcus. Essentially, coagulase is a virulence factor that's found in Staph aureus and a few other species of staph, but it's not found in most of the normal coagulase negative staph that live on and in our bodies. The reason that this is a useful test to know is that it's a simple and fast phenotypic test. It's not 100% reliable, but it's pretty good. Essentially, what you can do is you can take some blood products and put them in tubes and then take your bacteria and mix it with it. And if you see that it actually coagulates it and makes a clot like at the bottom of this B tube here, then it probably is Staph aureus. Similarly, we can also do it on a slide to see if we get a bit of this clumping or clotting activity. So when you hear people talk about coagulase negative staph, they're specifically meaning it to talk about staph that are not Staph aureus, because Staph aureus would be coagulase positive. So other than Staph aureus and coagulase negative staph, there are a few other staph that kind of are in between, and Staph lugdunensis is one of these. Staph lugdunensis is kind of a nasty guy. It fits an ecological niche similar to coagulase negative staph. So it does live on our skin and it can be a contaminant in a blood culture, but it also can be more aggressive and cause these more severe infections, such as cellulitis or causing endocarditis. So, Staph leishmaniensis is not that common of an organism, but I really wanted to bring it up here to you guys because I think it's important to understand that it's not just this binary dichotomy between nasty Staph aureus and not so nasty coagulase negative staph. There are other organisms like Staph leishmaniensis that fall a bit in between the spectrum. So when you have a patient that has gram-positive cocci in groups in one of their cultures, I don't think you can safely say, oh, this is definitely contaminant or, oh, this is definitely real. No matter what the species is, you do have to put your thinking cap on and think, how does this fit in my patient's clinical syndrome? So that's the first half of this talk. So tune into the next video where we'll talk about Streptococcus and Enterococcus.